Hello everyone, welcome to Antibody Engineering and Therapeutics Digital Week, brought to you by the producers of the face-to-face antibody engineering conferences, visiting Amsterdam and San Diego later this year. My name's Eric Warner. I'll be your host for today's session titled High Resolution Protein and Antibody Modeling with NovaFold and NovaFold Antibody. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other technical issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. All sessions are being recorded. You'll be emailed in 24 hours when they are available for viewing. Now let's begin by introducing today's speakers, all four of which are from DNASTAR. Dr. Steve Darnell, Principal Scientist, Matt Larson, Senior Software Developer, Thomas Leary, Manager of Sales and Technical Support, and Ho-Sung Shin, Director of Marketing. Thank you all for joining us today, and thank you to DNASTAR for sponsoring the event. Now we'll hand it over to our expert speakers.
Thank you for your kind introduction. Hello, thank you for joining us. Today's DNASTAR presentation is on high-resolution protein and antibody modeling with NovaFold and NovaFold Antibody. My name is Ho-Sung Shin, Director of Marketing at DNASTAR, and I will be your webinar moderator. Here is our roadmap for today's session. First, we'll talk about who is DNASTAR. The second, we'll move on to the introduction of the Nova tools for protein and antibody structure prediction. And then we'll talk about, how the modeling and the docking works in terms of science of it. And then we'll finish off with the live software demo with Thomas Leary. So DNASTAR is a pioneer in life science software. We started in 1984 by Dr. Frederick Blattner, who was a professor of genetics at the University of Wisconsin. And since then, we have added to our molecular biology suite with Genomics about 10 years ago, and shortly after with structural biology. One of the key focus for our company is to creating a connection between these domains to create workflows that are not available anywhere else. One such workflow is our SNP to structure, which we introduced a few months ago. It's a complete analysis from genomics to protein structure. And we think it's quite unique in terms of how we approach this market. So how does it work? Well, we assemble sequence data in software and identify variations. We take that and we direct access to common public data banks. And then from that, we generate structural models from the sequence. And we also simulate docking with other proteins. Now, Dr. Steve Darnell will take over.
Well, thank you, Ho-Sung, and it's wonderful to be able to talk to everyone. My name is Steve Darnell, and I'm a principal scientist here at DNASTAR. I'm also the structural biology team leader. We started the structural biology suite, as Ho-Sung alluded to, about 8 years ago, and we've covered a lot of ground since then. We started with our first application, which is Protein 3D. It's a molecular viewer of three-dimensional protein and nucleic acid structures. But we went a little bit beyond that. Besides just having a simple viewer, we also have the capability of doing sequence bioinformatics in the application. We have some structural analyses. We're able to create substitution variations in the application. But we knew that we need to go a little bit further than just that sort of analysis. And so we started developing what we're calling our line of Nova applications. These programs have either been constructed in collaboration with collaborators and partners, or we've developed some of the workflows completely in-house as well. The three that we're going to be talking about today are NovaFold, which is our structure prediction software application. Our protein structure prediction application. We have NovaFold Antibody for predicting the structures of different antibody constructs, and NovaDock, which is our flexible protein-protein docking algorithm. for those who are familiar with docking, please don't get this confused with small molecule docking. We are actually focused on these large interactions between full protein chains. So as we've talked with customers over the years, sometimes what comes up is that people don't really understand what the power of having a protein structure is and how they can use it to guide their research. So for example, you can use protein structures for trying to predict and characterize conformational B-cell epitopes. You can do that using linear B-cell epitope approximations and prediction methods like we have in Protein 3D. And you can try to map where those different linear epitopes lie within a protein structure. And you may see something like in the lower right where two linear epitopes seem to actually have a conformational connectivity. But you can also use our Nova applications for directly modeling predictions of conformational B-cell epitopes by taking a protein structure of your antigen, as well as using our software for predicting an antibody fragment structure. And then you can use NovaDock, and you can actually dock these two together. And the actual poses that are generated from the docking process are direct conformational B-cell epitope predictions. Additionally, you have the ability to start to model and characterize observed natural variations. Either we help at DNASTAR with people who have genomic data for trying to identify natural variants, as well as you can take this your variation annotations from any public resource like UniProt, and you can start to model those variations within the fold of your protein structure as well. And Thomas in our software demonstration is going to show that in action, but you have the ability to start to try to understand the energetic effects of what a variation will cause in the protein fold. And in addition, you can use a structure to help assist you in different protein design projects like humanization. While you may have your own tools for trying to reduce the antigenicity of a protein sequence, you also want to keep in mind the effect of the structural stability that those mutations make, and you can start to probe those sorts of questions using Protein 3D, DNASTAR structural biology suite, and a protein structure. So we are highlighting our Nova applications today. I'm going to start off with NovaFold, and this is our general purpose structure prediction program. NovaFold was developed with a strong partnership with Dr. Yan Zhang from the University of Michigan. He is the principal investigator of the iTaser algorithm, which is a highly accurate hybrid-styled approach to doing structure prediction. DNASTAR is the exclusive commercial vendor of the iTaser algorithm. We're very proud of that responsibility, and we take it very seriously. So when I mentioned before that iTaser is a hybrid approach, it goes beyond just homology modeling style approaches. So rather than just trying to do sequence comparisons with known experimental structures, the hybrid approach is that it uses a technique called protein threading to perform pairwise alignments using not just sequence, but inferred structural characteristics like secondary structure, solvent accessibility, and internal internal residue contacts. And in addition, there are ab initio style techniques that are used in order to perturb those starting positions identified by those templates and to deform the structure into lower energy conformations that are more appropriate for the query sequence that you give it. Again, after a structure prediction, it's actually quite simple to start any of these. All you have to do is provide us with a protein sequence. You have the option to control some of the parameters of the application. But it's a fully automated pipeline that will deliver a detailed report. And we'll go through that, or at least overview that in the next few slides. But it's an interactive report in Protein 3D that's intended to help guide you through the analysis of your structure prediction. And in addition, something unique to the iTaser algorithm and NovaFold is we have the ability to take those models and do a structural analysis for you to try to predict protein function using a combination of techniques. So the iTaser algorithm itself has been benchmarked over the past decade in an experiment called the Critical Assessment of Structure Prediction, or CASP. The ITASER server has been, or is listed as the Zong server in the CASP competition. And for the past 12 years, it has been the #1 ranking automatic algorithm available. The Z-scores that you're seeing are actually a significant score describing ITASER's performance compared to the rest of the field. And there are a number of other algorithms that you may have seen as well. And ITASER, though, is just a very strong and robust algorithm that works for a very large array of problems. So NovaFold, when it's completed, will provide a visual report that you can view in Protein 3D, and you'll get up to 5 models, our best predictions of what the fold of your protein sequence is. There will be some quality metrics associated with it, including a template model score, a TM score, and a root mean squared deviation, an RMSD. These are standard metrics that are used to characterize structural similarity between 2 superimposed structures. Now, since this is a structure prediction, we don't actually know what the real answer is. So using a regression model, we are predicting what the TM score and RMSD of this protein model would be if the true answer were known. TM score is a normalized score between 0 and 1. 1 is best, 0 is worst. And RMSD is a distance metric that actually we're trying to minimize that. So a value of 0 means that there is no difference between models. So lower scores in RMSD are better. So in addition to that overview, you have the ability to take a look at the templates that were selected from the protein threading algorithms. We provide little mini maps that are color-coded to help guide both the coverage and the % identity associated with the template selected, and a little map to help identify what regions of your sequence were covered. You have the option to drill down a little further with our report. If you are interested in seeing the residue level sequence alignment of your templates against the selected templates against a query sequence, you can open them with one click in Megaline Pro, our multiple sequence alignment program, and that's displayed in the lower right-hand corner. Additionally, part of the process of a NovaFold prediction is that we collect similar experimental structures related to your model. So when you are trying to predict protein function, we will take your model and we'll do a reverse structural alignment search throughout the protein data bank. And that list of structures in the lower left-hand corner are real structural alignments between your model and known experimental structures. We use that collective set of known structures to help guide our function prediction. So it's not sequence-based approaches to predicting function. We use structural approaches. They can be used to help identify binding pockets and enzymatic function, as well as we have a machine learning model that is designed to predict Go terms to represent protein function. And again, those selected experimental structures are used as scaffolds to look for through a curated data set of known small molecule binding sites. And that set of structures is used to identify conserved or at least high confident small molecule binding sites. And in the report, you'll actually see the binding site residues associated in your protein structure, as well as a rank order of different types of ligands that may be associated with your model. So we're going to talk next about antibody modeling, and for that I'm going to pass the presentation over to Dr. Matt Larson. He is a senior developer at DNASTAR, and he is the lead developer for the NovaFold antibody algorithm.
All right. In addition to the general structure prediction software NovaFold that we were just discussing, in the last 2 years, we also released NovaFold Antibody, which is a specialized program for multiple chain antibody structure prediction. NovaFold Antibody can start from amino acid sequences and predict the Fab, Fe, light and heavy chain structures. It will automatically annotate major features such as the CDR loop sequences and can predict CDR loop structures via multiple approaches, including from a canonical loop library and ab initio methods for the H3 loop, and also using a machine learning approach that we'll discuss next. Shown on the right side here is the general pipeline, where we start with the search for the templates from a large library of antibody template structures. After we find the best light and heavy template structures, we graft on CDR loops using one of the 3 different approaches. After that, we minimize and finally return 1 or more structures, depending on how many structures you want to pursue. For the machine learning approach, we're looking at majorly at the H3 loop, which is the hardest of the 6 CDR loops to predict. And what we've implemented in NovaFold Antibody is an approach that can use both the sequence of the H3 loop for this particular query structure, as well as the geometry of the stem. And using both of those features, we can find the best loops from a large dictionary of over 300,000 loop structures that have been mined from the protein data bank. And we can actually return that as an ensemble when you want multiple structures. What we return at the end is an annotated report, which includes both predictions and lists of what is the highest likelihood heavy chain templates, highest likelihood light chain templates, annotations for the locations of the CDR loops, and all that can be easily imported in a Protean 3D. And inside of Protean 3D, we can actually quickly click on a feature and display and highlight the CDR loops and focus on that region, which is often the major focus of an antibody structure prediction. In addition to large internal benchmarks that we've run with hundreds of structures, we've also compared the NovaFold antibody structure predictions versus one of the more recent, smaller benchmarks that compare different companies' software prediction packages. And in this benchmark of about 11 targets, we predicted the H3 loops with average RMSDs of 2.3 angstrom RMSD, which is right at the top. One of the advantages of the NovaFold antibody structure prediction is coming from fragments, we can actually do this process very quickly. With a local installation on a Linux machine, we can make the predictions within 5 minutes. So that's one of the nice things, is we can provide high accuracy CDR predictions and do them quickly. Shown here is the breakdown from this benchmark for 11 structures, in which the NovaFold antibody is shown in blue. Lower RMSD scores are better. And on average, we're doing better on most of the targets shown here, and that's what led to a nice low RMSD for an average. One of the things that we like to do with antibodies is also see how they bind. And so I'm going to pass back to Dr. Darnell, and he's going to go on further with our docking software.
Thank you, Matt. So the third application, Nova application we're going to discuss today is NovaDock. And as I said before, this is our flexible protein-protein docking program. So for those who are familiar with small molecule docking, this is really not that much different, except for that both the ligand and the receptor are protein structures, macromolecules. We're going to be talking in the context of antibody-antigen binding for this presentation, but NovaDock is a general purpose tool as well. And it can be used to dock any pair of protein structures that you have. Each individual binding group, either the receptor or the ligand, can consist of multiple chains, much like an antibody. So what we're interested, of course, with docking is predicting how the receptor and the ligand are going to bind with one another. And what NovaDock will do is to spatially arrange a ligand against a receptor structure, both through three-dimensional space with translations and rotations, but also with a flexible modeling approach that deforms the protein backbone along a mathematical model called normal modes of motion. So NovaDock was constructed in partnership with Dr. Paul Bates at the Francis Crick Institute, and it is powered by the SwarmDock algorithm, which is a high-resolution docking algorithm that uses a minimization approach called the particle swarm optimization. I'll talk about that in just a minute. But SwarmDock is again has been benchmarked against the leaders of the docking field in a competition called CAPRI, the Critical Assessment of Protein Recognition. It is actually SwarmDock in the last competition was a top three docking tool, and doing very well and providing actually for the first time in that competition a very broad approach to modeling protein flexibility. Now, that flexibility is actually captured, again, as mentioned, through these normal modes of motion. They are a mathematical model that tries to describe large concerted changes through the entire protein fold along resonant frequencies. And they can be combined and sampled in a very exhaustive way. So rather than just flipping side chains like many flexible docking programs do, we are perturbing the structure internally as well. The algorithm itself is being represented by that animation in the lower right-hand corner. So if you imagine that large red sphere is actually the receptor, and all around it is going to be hundreds of different particles, in this case copies of the ligand. And when this animation restarts itself in a second, this is just one small slice of a local search. What's going to happen is that there's going to be hundreds of particles that are initialized, and they're going to start to sample the structure like this. And the swarm communicates with one another, and there is basically a leader of the swarm, and iteratively the molecules try to look more like the leader until they converge onto a solution. We perform this sampling across the entire protein surface, collect the results, and cluster them in order to present our final docking models. And as with NovaFold, Protean 3D can display a NovaDock report that includes an overview section, as seen in the upper left-hand corner. The central molecule is the receptor, and all those spheres represent poses of different ligand presentations. Each sphere is color-coded by an energy score. Green is lower energy, red is higher energy. We're interested in the lower scores. And additionally, as I mentioned, there's a clustering going on, so larger cluster sizes often are correlated with lower energy, more confident models. So the spheres are also sized, the radius is scaled depending on their cluster size. So what we're often interested in is larger spheres or green-colored spheres and those that cluster together. So in addition to being able to just see these models at this level, you can drill down a little lower. Our report has a full atomistic scale model where you can actually look at the atomic interactions in a little interactive thumbnail. There's also a table describing all of the intermolecular contacts that every residue in the protein interface is making. Those are interactive, and you can control the visibility and selection. And if you choose, you can actually explode those models into full protein structure views, and you can explore your protein model, or protein-protein docking results with all the capabilities within Protean 3D.
Thank you, Dr. Darnell. Just beautiful. I think we're quite proud of what we built here. Not only how we visualize these models, but also the level of sophistication we go into presenting the data itself. We felt like our platform is well integrated, easy to use for researchers. You're able to run predictions either locally or on the cloud, so it gives that flexibility. And the integration of within the structural biology suite, as well as with our genomics and our molecular biology suite, is an added competitive advantage for us. And best of all, we find that it's quite easy to learn and easy to use. And in cases where that may not be the case, we provide great access to what we consider world-class technical support and scientific staff. Now here is our domain expert, Mr. Thomas Leary to actually do the live demo.
Thank you, Hoson. Thank you. So I'm one of the trainers here at DNASTAR. I'm one of the people that you can call up essentially anytime after 7 a.m. U.S. Central Time till 7 p.m. Central Time and get help on-the-fly webinars, things like that. We can provide on-the-fly training whenever needed. Now, as Hosung mentioned, this suite is really comprised of the three domains of life sciences: molecular biology, genomics, and structural biology. Now, this application right here is really the hub for all the different tools and then any information you need about them. As I hover over each of these items, it describes what the tool can do, and also there's videos and training and tutorial tips that you can use to learn the suite. So if we go through the molecular biology tools, we have SeqBuilder Pro for cloning, PCR, primer design, general annotation. Additionally, it's got auto annotation capabilities where we can auto annotate plasmids or bacterial strains from other strains that you may have. Also Megaline Pro for multiple sequence alignments. SeqMan Pro for Sanger assembly and variant detection using Sanger assemblies data. SeqMan and Gen for NGS data, Illumina assemblies, targeted exomes, RNA-seq, ChIP-seq. ArrayStar, which can be used for multi-sample variant detection analysis, as well as differential expression experiments using RNA-seq data. And then here we get into the structural biology suite. But quickly, as SeqBuilder Pro does relate quite to the domain of structural biology, I just wanted to quickly show you this tool. It's a new interface we have. This has been updated to 64-bit capability as of the 15.1 release. And this is a general tool that you can use for primer design, cloning methods, reverse complementation of sequences. Also, we can look for enzymes. We can make restriction digest maps of enzymes that may cut, and then also have access to a very powerful auto annotation capability. I just wanted to quickly show you this because this is one of the tools that really can be used while working with proteins, and it's related in that way. But then also, as we move on, I'm going to highlight Protein 3D's capability and how we can get access to the Nova Cloud services, and then also we'll jump into some variance mutation analysis. So here is the Nova Cloud services pane, and so I'm going to click here. I've already logged in, and as you can see, we have a number of predictions that have already been run. But what we can do is run predictions directly from this interface, and we have access to NovaFold, NovaFold Antibody, and NovaDock. So I'm going to click on NovaFold Antibody and kind of overview the general use of this tool. So here it's asking for me to add a file of the antibody, the heavy and the light chain. So if we take a look at this file quickly, you can see that it's an annotated heavy and light chain file. It doesn't actually matter if it's annotated or not. The minimum Markov method will determine the heavy and the light chain for us and then annotate it appropriately. Also, this could be a single chain if needed, and so it can handle single chain as well. So I will go ahead and select that, and then once it's inputted here, you could see that the number of residues is specified, and then also the sequence itself. We do have additional options. For instance, we can refine the heavy and light chain variable regions. We can also specify an increased number of H3 loops to model. Additionally, we can supply PDB files that can be used to guide the assembly or the structure prediction of this. Also, we can exclude different chains. So there's a number of options, and in the local version, there's even greater number of options. So actually, I'm going to go ahead and submit this job to the cloud directly in real time, It'll be running as we overview this. Now, NovaDock has a similar interface where we can supply PDB files that we want to do a protein-protein docking with. So here I will specify a receptor, PDB file, and a ligand. And then also we have additional advanced options. Steve was talking about the number of normal mode perturbations that are applied to these structures to look for internal variations that can occur along a mathematical predicted normal mode. And we can increase or decrease the amount of normal mode perturbations that are occurring in both the ligand and the receptor. Also, we can specify residue contacts for both the receptor and the ligand. Now, if those are known, we can also do blind docking if those are not known. Additionally, the NovaFold prediction will give us some binding interface information. If we have that information, we can use that here to direct the binding or the swarm to those regions. I'm actually not going to submit this one, but we're going to go back, and some of these predictions have already been run. You'll notice this is the one we just loaded in there. It's currently running. It's got a 3-minute elapsed time. Also, to take note here, the antibody predictions in the cloud do take 30 minutes or so. This is due to some spool up time that we have. The local version does take around 5 minutes. I can access these predictions directly with a one-click open here, and then we can get access to the model. It'll download, and I can start interrogating the model. Now, there's different tools we can use to view the model here. Additionally, we can start looking at the hypervariable regions quickly by clicking on the hypervariable tag, and I can color these regions if needed, a different color. And if we want to go in further, we can start looking at just the H3 loop and then color it separately if needed for annotation purposes. Like that. Additionally, we can export this file to a PDB file directly and then use it in subsequent NovaDock predictions. Now, I'm going to jump back to this Nova Cloud Services pane here, and I want to show you a NovaDock prediction that was run through this pipeline. If you notice here, it does give us some additional information, a summary statement about the interaction. So here's the model overview. As you can see, Steve was pointing out the spheres and the coloring. The spheres indicate the size of the cluster, and the coloring indicates the level of energetics. So green is lower energy, red is higher. Now, interestingly, this third model has a cluster size that's actually greater than the lowest energy model, which is interesting to us because although we found a lower energy conformation, we also found one of slightly higher energy, but with a much larger cluster size. And if you notice, all of these are within a similar region. So what I'm going to do is open up this third model because that's the one I'm most interested in. And I can then see the residue interaction between the receptor and the ligand surfaces with the intermolecular contacts. Now, this is also displayed in a cloud interface. It's not required to use Protean 3D for these models. We can submit them directly through Nova Cloud Services through this web interface and then access these models directly. And so we'll go to the model we were just looking at, and there will be a nice textual report of the interface and then also the log of the run. So this is useful in case you want to use the web services to access the cloud instead of the Protean 3D application. We can then download this from here and load it into Protean 3D or share it with our colleagues. So let's go, and I'm going to actually open this model. We can manipulate it directly in this small preview window, but I'm going to open it up in a large new document. So this is the PDB, the two PDB files interface surface right here, and then we can actually start interrogating more closely this interface. Also, if we go to the analysis pane, you can then see receptor contacts are listed, so that you can kind of have an annotation of them. Now, one of the tools that we can use for this interface is we can start applying residue variations to these residues. So I'm actually going to select this residue. And then what I can do is go to the structure, create variant, and I can apply a substitution to this variant. So first thing I'll do is add a tryptophan, and then look at the energetics and the side chain positioning. So it looks like we actually had a rather large, anything larger than 1 d-fire energy change we consider significant. Anything below 1, we would possibly say that it's significant. Dr. Larson can go into that later with our next prediction. But so here we had a residue variant that had a negative 1.129 d-fire energy change. So we could say possibly this was a stabilizing change. Also, it'll apply the new energy minimized side chain here, and we can start looking for clashing or some sort of other steric disruption that occurred by adding such a large group to this residue. Additionally, I want to show you a substitution of a proline. And if you notice, as expected, due to proline's inability to bend or flex, we had a large increase in d-fire energy, 2.329. Dr. Larson, what do you think about a change of that size?
This would most likely be a destabilizing mutation. So this would be one that is detrimental to the structure's conformation.
Okay. And then we can also apply the side chain to the residue and then start interrogating it for clashing. If you notice, the 3 side chains are displayed here, yellow, blue, and red. So the original and then the 2 new variations we've applied. And those can be hidden if needed. But they're there so that you can kind of investigate the different lowest energy positions for these side chains. Now, one of the things we've done, and one of the things that Ho-Sung has mentioned, is that we have this new SNP to structure workflow. You can do this sort of one-off structure variant mutation analysis in Protean 3D, but what we've done is we've integrated with our NGS tools the ability to do the same type of thing. So what you're looking at here is you're looking at a exome study, a targeted gene panel study of paired lung cancer versus normal tissue from Illumina data types. Now we've ran this through our NGS assembler, which is this right here, SeqMan and Gen, and then we've opened the project in ArrayStar, which is for multi-sample filtration. And what I can do here is we can send this position over, this Llama 5, we can send these positions that we're interested in over to SeqMan Pro so that we can interrogate the differences. But quickly, I wanted to show you, ArrayStar has a very vast capability for filtering and looking for different variations that meet statistical criteria that we're interested in. For instance, we can look for non-synonymous substitutions, different types of non-synonymous substitutions. Also, we can specify depths and SNP% metrics. We can apply 1,000 genomes, population genetic data, functional prediction data, evolutionary conservation, and pathogenicity data into our filtration. So we can really hone in on SNPs that have known clinical significance to a disease model that we're interested in reviewing. So from here, I can send this Llama 5 directly over to SeqMan Pro, and we can start reviewing the actual reads that make up this assembly. So actually, one of the things I like to do is sort the reads to the variant. So what I can do is select the region in the consensus and then sort them. And then you see that the reads that made up this call can be grouped together. And so you can really visualize how we made this call with the data and the reads that actually present this information to us. Also here we see that we have a PDB ID available for this Llama 5 isoform right here, indicated by this square bracket 1. And I can send this over to Protean 3D. What it's doing here is it's scanning through the PDB files that are available and determining the best structure with the lowest angstrom resolution to use for applying the residue variation. It will then do so automatically in this automated workflow, and then present to you the sidechain energy minimized and then the D-fire energy from this this mutation that we've detected with our NGS data. Now, Dr. Larson, this is a 1.058 D-fire energy. What do you think about this?
And again, this kind of category, if it's slightly over 1, I would say this is most likely a destabilizing mutation, which would match with the variant that we're looking at, which is going from something that's basically some hydrophobic properties to something that's of a very different class, the cysteine residue. So yeah, that seems to be a slightly destabilizing mutation.
And then additionally, it's interesting that it is on a surface accessible portion. So those two pieces of information are rather interesting. Additionally, we can look at this other PDB ID, which is a different variation at a different residue. And then see how that energetics change with the residue variant that we found with the NGS data. And so this one appears to have a negative 0.5. Looks like this is possibly stabilizing. It's below our absolute value of 1 change that we would say is significantly destabilizing or stabilizing. But how would you gain insight from a negative 0.5 like this, Matt?
This looks like it's on the side of what I would say you could say is stabilizing. An additional nice feature that will be coming out in 15.2 is that the table that shows these mutations will actually have labels to help people know if it's stabilizing or destabilizing as well.
Perfect. Thank you. Yeah, so I guess that's a quick overview of kind of how we've integrated the NGS workflows and the structure prediction workflows. Obviously, these NGS workflows can be rather complicated, and that's one of my roles, is where I can help really people learn these tools if you're unfamiliar with it, and then learn how to use them and use them with your structure prediction. Now, one of the things that could help you is this human genome nomenclature format that we provide. It gives you a concise residue mutation format for you to see which residues were changed and at what position. So this is quite helpful. And if any of you guys need more information on some of the NGS tools or the molecular biology tools that I showed today, I would be happy to present another personal webinar to anyone interested in learning more about those types of tools. But thank you guys, and that kind of concludes my presentation, my live demo.
Excellent. Thank you, Thomas. That concludes the prepared portion of our presentation. We hope you enjoy the material and the tools that were showcased. We're honored to have almost 55,000 users around the world at thousands of universities, research institutions, and corporations. If you have an interest in our products or services, we would love to discuss your area of interest on a one-on-one basis. Please send us a message to this email address shown here and mention your attendance at this digital conference. Thank you. Now we will hand it over to our...
Thank you all for an excellent presentation. We've received a few questions already, but we'll give the rest of you a moment to enter your questions in the Q&A box to the left of the slides. Before we begin the Q&A, I'll run through some quick announcements. First, I'd like to again thank DNASTAR for sponsoring Digital Week. And next, I'd like to quickly draw your attention to our face-to-face antibody engineering and therapeutics events. They're visiting Amsterdam June 5th to 7th and San Diego December 9th to 13th. At these market-leading annual events, you'll have the opportunity to meet experts face-to-face and network with hundreds of peers. Now let's hand it back to our speakers to begin the Q&A.
Well, thank you very much. I think we're going to start with a question that we got from all you listening with regard about local computing. So the question is, can I run predictions on a local machine if I don't want my data to be hosted in the cloud?
Well, Steve, we can run the Nova local applications on a system that supports Docker. And so currently we are supporting Linux, but we can also do the installs on Macintosh computers that have Docker installed with a few slight modifications. And then also, because this is sort of Linux terminal kind of setup, you will have access to talk with me or a webinar to help with the installation of the command line tools.
All right, thanks, Tom. We have our second question so far, and it's about our machine learning methods. So you mentioned machine learning methods. Can you tell us more about how the software uses those? Matt, do you think you take that one for us?
Sure. In particular, in the NovaFold Antibody software, there's at least two places that we're relying heavily on machine learning models. One is the selection of the framework regions, the templates that are composing the frameworks for the antibodies, where we've built a large hidden Markov model library for about 2,600 known antibody structures. In addition to that, the threader, which actually searches through this library, then is passing through the sequence through the hidden Markov models to find the best least likelihood scores and z-scores, which contribute to choosing which framework to use. And the hidden Markov model-based methods have consistently been the best methods for protein threaders for actually finding templates. The other place that we rely heavily on machine learning models in the NovaFold Antibody program is for choosing the hardest of the loops, which is the H3 loop. While the other 5 loops we can actually consult a canonical loop library and use the sequence mostly to choose those loops. The H3 loops, because they're large length, that allows for lots more of loop confirmations than the other smaller loops. And because of that, what we've relied on here is we rely on a random forest model, which is basically a decision tree or mini decision trees put together. And so then we can have lots of parameters such as the stem geometry, the sequences, distance between the endpoints of the loop, and all that. We don't have to worry about which is some important parameters. We can toss that into the machine learning model with a large library of loops where we have about 300,000 loops that can be used with all their different parameters. And the random forest will actually find a way that, in a non-biased way, to assess which parameters are the most important for choosing loops. And then this will end up being often the stem geometry and the sequence. So that's one of the major places that we've really taken advantage of, and that's really helped with finding the best H3 loops for the NovaFold Antibody software.
Thanks, Matt. This next question has to do with kind of like what's required by NovaDock, more like with sort of inputs it takes. So it's how close should the ligand and the protein interaction be before you can use NovaDock, and also how much information about the interaction do you need before performing a NovaDock modeling? I can take that one. NovaDock kind of operates in two particular modes. One is directed docking and one is blind docking. So when you provide input structures for docking, it's not really refinement like other tools where they have to be very close and near the final solution. what you can do is you just provide two, a binding partner, a PDB file for a first binding partner and a PDB file for the second one, and blind docking mode will basically try to sample the entire surface of the receptor that you choose and try to provide poses that make sense. The kind of the restricted docking mode, if you happen to have more information regarding which residues you think are involved in binding, you can tell NovaDock to focus more on those. So one example that we use all the time around here is with antibody and antigen docking. We tend to put the antibody as the receptor and just mark the H3 or the CDR loops as the receptor interface. And so NovaDock will just focus and only sample around that area. It's still kind of doing its same blind-like search. It's just very restricted in the area. So it's not NovaDock in itself is like one of these. It's a docking program that isn't really used for like close refinement, but for helping to discover new poses, and we rely on the algorithm kind of settle down and minimize on that. Another question in regards to the people who are actually using the Nova application. So are there any commercial antibody companies using this software? Pardon me. Tom, could you take that?
Yes, Steve. So we do have a few of the top 10 big pharma companies using the Nova suite.
Okay.
I can't go into the details of.
Sure, I'm sure everyone understands. I do also know too. I kind of watch the literature, and there are, certainly some of our tools are actually newer, like NovaFold Antibody and NovaFold, and compared to our other DNASTAR applications. We currently have dozens of citations that have occurred in the past 1 to 2 years as well. So those are easily searchable for as well. Our next question is about in silico humanization. So can this software be used for humanization on mouse antibody sequences? Yeah, I could do. I'll answer that. I think at this stage, the structural biology suite can assist in it, but it's not necessarily designed to perform humanization workflows yet. And what I mean by that is we currently don't have tools that are designed to kind of search for like structure-based or other sorts of more modern immunogenicity metric. Now, Protein 3D does have a number of classical approaches for T-cell epitope prediction, and B-cell, and one for that we constructed about 2 or 3 years ago for linear B-cell epitope prediction from sequences. But you kind of have to use all the pieces together on your own. I think where the structural biology suite is really useful would be more like if you have your own de-immunization protocols that you're using, but you're interested in knowing whether or not you're going to be stabilizing the fold of your antibodies themselves, which is, of course, another important thing. If the antibody doesn't hold the fold that you expected it to from the mouse model, then it's going to be more than likely inactive and not as useful as well. So you could use the capabilities in Protein 3D to help kind of screen as another filter beyond that point too, to look to see and to rank your possible mutations by predicted changes in energy.
Can I add on to that too?
Please do.
In addition to kind of the direct structural workflows, we have lots of nice tools in our laser gene suite, such as Seq Builder, where you can do kind of like sequence editing and tools like that. In the case of trying to humanize an antibody, one thing you could try to do is pass it through the NovaFold Antibody and get the annotations for what the loop sequences are, and then take a template, such as a human template, and basically graft on the loop sequences into the human sequence. So you'd replace the loop sequences from the human one with the ones from the mouse antibody. So you could try some custom approaches like that. using multiple different tools, but we really don't have like a fully automated pathway for doing that right now.
All right, so it looks like that's all the time we have for questions today. Steve, Matt, Thomas, Hosung, thank you all for a great session.
Thank you very much, and thanks for everyone who has made it to the end, and we appreciate the time you've taken out of your days for us.
Thank you.
Excellent. And everyone, be sure to download the presentation slides in the resource box, and we'll send you a reminder when the recording becomes available. And before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Connect 365 Life Sciences, have a great day.